I would also like to thank Dr. Nikki Malhotra, Mr. Jeff Lewis, and Dr. Charles Hinley for their support, advice, and guidance throughout my project. And there's some stuff. Yeah.
Thank you, Bronte. Okay, so can you tell me how the azithromycin really inhibit? How does it inhibit the bacterial growth?
Azithromycin, as a general antibiotic, attacks the cell membrane of bacteria, and it has been recently, from other scientific papers, I have found that it is a quorum-sensing inhibitor for Pseudomonas bacteria, and in particular Pseudomonas aeruginosa, which is a part of surgical site infection.
Okay, so then you added the hemiltenin, and
Yes.
that further increased the effect?
Yes,
The
it
synergistic
increased the
effect?
antibiotic.
So how do you think that is improving the inhibition?
So since azithromycin worked as a general antibiotic, the hemamilitanin, it started to inhibit the quorum sensing within Staphylococcus bacteria, and I tested on Staphylococcus bacteria, so I really got to see the difference between azithromycin alone and azithromycin with a possible quorum sensing inhibitor for Staphylococcus.
So is it just a general thing, or is there a model? Are you proposing something? How is hemiltenin really increasing the inhibition?
Well, seen from the zones, there is a greater measurement of zones of inhibition with
But why? At the cellular and molecular level? Why do you think it's...
Well, hemamilitanin, as I mentioned before, is it interrupts the quorum sensing of Staphylococcus bacteria, specifically with the RNA3 enhancement within that process.
Okay, I wanted you to get into a little more detail.
Okay.
Okay, another question. I remember that, before doing your actual work, you waited a long time.
Yes.
Can you explain that? Why were you waiting for that long?
Okay, so basically before I continued with my project, there was materials that went into it that I wasn't sure if I wanted to use and how much I wanted to use. And since a lot of these antibiotics, specifically hemamilitanin, have not yet been fully studied, I didn't know how much of that drug that I needed to order. So I had to take time to figure out exactly how much I thought would be best for this study. And I also was basically intimidated by the fact that I didn't have that much time left. And then with bacterial growth, like if anyone has ever worked with bacteria, they know that there's time limits with every step of the project. For this specifically, I had to work in 24-hour increments. So it was mainly just scheduling and planning.
And then you waited even more?
No. Then it just took me time to order the bacteria and get it here. I also was planning on working with more bacteria in the beginning than I ended up working with. However, I think it worked out better to start with something small and then to enlarge it later.
Okay. I am going to be sending you to AP seminar kids.
Okay.
You're going to go into their class and prepare them a little bit for next year. So what are you going to tell them?
I'll tell them not to waste time, because time, even though you can't buy it, it's the most expensive material in scientific research. And I thought that I wasted a lot of it, and now I know for future scientific research opportunities to not waste time and to just get started and not be afraid of the results. Because any result in science is an advancement.
Everything is significant. Thank you.
I think most scientists are procrastinators. At least the ones I know. I just wanted to follow up on that mechanism of action for azithromycin. Is it inhibit cell membrane, or does it inhibit cell wall synthesis?
I found through my research that it inhibits the cell membrane itself. Yeah.
Do you know if it is does it poke holes in the membrane and it just allows everything to leak out?
Yeah, I found from the
Campus security, please call the activities office. Campus security, please call activities office.
Okay, that's good.
From the research that I found, it always described it as attacking the cell wall, so I'm assuming that it did poke holes in it and diffuse it that way.
Okay. If hemamilitanin is a RNA RNA polymerase III inhibitor,
Mhm.
and you didn't see any effect on inhibition of growth. Do you know, were you using the right concentrations for your experiment?
So with hemamulitanin, there wasn't that many studies out there that used it with a wide range of antibiotics, so I didn't know exactly how much I would need. So that's why my concentration gradient was larger between when you compare my azithromycin concentration gradient and my hemamulitanin gradient. And it was mainly just an opportunity of having the materials, and I had a total of 5 milligrams, and then when I made my stock solution, I had to really focus on how I could transfer that much of the powder stock into the water to make sure that it was the right solution and I had enough to complete multiple trials.
If you had to do the experiment over, what would you change?
I would increase the concentration of hemamulitanin as much as I could because there is
Until you saw a...
Until I saw a zone,
Yeah.
yeah.
Okay, good. Okay, that answers 2 questions. Let's see. Why do you think you saw a decrease in efficacy upon increases in concentrations?
After researching other scientific papers, I've seen that with antibiotics, there's generally a rise and then there's a peak and then there's not much after that peak that can improve. So when I was looking at the peaks, most of them would rise and then at that point the bacteria that died would die and any major decrease could possibly be of human error or of just the bacteria's ability to reproduce.
Let's see. I have another question in here. Yeah, I guess how do you think they synergize with each other? Is it just
it's
2
an
different
additive.
mechanisms of action coming together?
I think that since azithromycin works on its own as an antibiotic and hemamulitanin works on its own as a quorum sensing inhibitor, I think that the 2 of them, it would be an additive combination that they inhibit the growth better together.
Okay. If it's an RNA polymerase III inhibitor, do you think there's genes that are expressed under RNA polymerase III that are specific to quorum sensing?
When I was researching, I saw a diagram of the quorum sensing aspects of Staphylococcus bacteria, and in one of them, if I remember correctly, it had RNA3, and then from there branched out possible biofilm formations and possible reproductive mechanisms.
Okay, thanks.
Thank you.
I wanted to ask, you took your best concentration of azithromycin, you added to the hemamulitanin. How come you didn't go the other way? How come you didn't add the hemamulitanin to the azithromycin? Because you could see the result with the azithromycin, but you couldn't see any result with hemamulitanin.
That's true. I think, as mentioned earlier, with the amount of concentrations that I had of hemamulitanin, I didn't know how to go about finding the best concentration to add azithromycin to the hemamulitanin with the given time restraints.
So you're sort of limited on ingredients.
Yes, mainly.
And then getting to your statistical analysis, you said that the diffusion was actually, in combination, was statistically different than...
Yes.
Oh, okay. Do you have a chart for that? Did you show a chart of that, the difference between...
I have a chart that has the difference between the zones for each concentration individually per...
Oh, hemamulitanin didn't have any charts, right?
No, because when I recorded it, there were no inhibition zones to be measured, except for the stop of growth of the disc itself.
Okay, so what did you compare it to then?
I compared for azithromycin.
When you did your analysis and you said that the combination was significantly different than the original. But if the original showed no disc formation, what did you compare it to?
I compared the additive to the azithromycin by itself. I turned the azithromycin, just the 1 microgram per milliliter of azithromycin. I compared the changes to that.
Oh, that was your basic, you're
Yeah.
comparing it.
I compared it to I used azithromycin as a control for the addition of hemamulitanin throughout the rest of the experiment.
Okay, and so that's where you're at, oh, that's where it was, okay, that's right, yeah, that was.
Yeah, no, I could see that, how that could be confusing.
Okay, alright, great, no, that's all I got, one, nice study.
Yeah, it's pretty cool.
Okay.
Okay, so we're waiting for Zach.
Zach's the same.
Jack Adams.
There's Jack.
I think he was just waiting outside so that he wouldn't interrupt.